Alrighty. Good morning, everybody. So we are going to pick up where we left off in our discussion. So we'll wrap up talking about DNA and RNA, protein synthesis, the central dogma, control of gene expression. And we're going to talk about a very straightforward topic today regarding genetics and inheritance. So pretty much when we review the stages of the central dogma, we have the DNA molecule, the original DNA molecule, which is then transcribed to the mRNA molecule, which then will become the protein. So remember that the proteins are basically what's causing an organism to look the way they do. So you are an expression of all your proteins. So whether it's eye color, hair color, skin color, so on and so forth, it is all the expression of your different proteins. So obviously the discussion is genetics, and boy, have we come a long way from when Mendel first discovered the idea that there are discrete units, which obviously could not describe because they didn't have the method, the modality, technology. Obviously, there's some units that were going to be passed along from the parent to the offspring. So before the 20th century, again, that was Mendel in the 1860s. Basically, Gregor Mendel was a mathematician who was a genius in his field, but he decided to drop all that and go and become a monk in a monastery. In that process, obviously, he had a lot of time on his hands. So there was a garden that he had at the monastery where he was basically planting different types of the pea plants. And he was able to discover two important processes. Basically, that the parents transmitted their discrete units to their offspring. But back then, back in the day, they thought that maybe it was a blending of the traits. But if it was a blending, then how come all the offspring that parents had look different? So why do siblings look so different from one another? So he was questioning that. But again, they also thought, which is kind of crazy, that's why I have this image here, that an actual baby, yes, was actually carried in the head of the sperm, which is obviously not true right now. We know that for a fact. The only thing that's carried in the head of the sperm is the genetic material in the area known as the acrosome. So he started asking that question, basically, why don't all siblings look alike? If parents were to have 10 kids, none of the kids look alike. Now, again, there's going to be similar features, obviously. I mean, I am one of five. There are similar features between my brothers and my sisters, but we don't look anything alike if we were to look at each other. So basically, Gregor Mendel started conducting this scientific experiment, and he used a very simple breed of organisms known as the pea plants. Because the pea plants could grow really quickly, he could actually separate the different traits and the individual traits of the pea plants, and they were actually he was able to fertilize them, so cross-fertilize them, and again, create different offspring. So basically, he was able to break it down into the pea plant color, the pea color itself, the pea size, the shape of the peas, the size of the stalk, the petals of the pea plant, and then the flowers that would bloom from those pea plants. So when we look at Mendelian genetics, so that's just everything that was discovered by Mendel, basically there are four main concepts. The fact that there is heredity, and that's the transmission of traits from one generation to the next. The fact that he's able to create and help to, not create obviously, but able to recognize and name the study of heredity as the field of genetics. He is also able to determine that there is what's known as character, And those are basically the heritable features that vary among the different individuals, and that there are also the different traits. So those are the variants of the characters. So basically what he did was he created the true breeding varieties of the plants to study the generation. And basically what he did was he took two parental generations. So if we look here, you have the dominant, which is the purple blooming flower, and the white, which is the recessive. And you can see all these other traits, which are dominant, and all the other traits right here, which are recessive, relating to the pea plant. Again, he was able to cross, and he was able to see different traits arise. So he crossed the P generation, then he crossed the two F generations, and he was in for a world of surprise when he did that. So the first hypothesis that Mendel had was there are what's known as alternative versions of the genes. He didn't call them genes because it hadn't been discovered yet. Again, remember, this is the 1860s. So basically, he said that there's something, units or something that's causing the pea plants to come out either purple or white flowers or yellow or green peas or the wrinkled or smooth peas. So he called them traits, he called them units. We know now they are genes. So we're using the definition that we have modified in modern genetics to say that it is alternative versions of genes which are going to account for the variations in inherited characteristics. So one, each gene can have multiple alleles. Remember the alleles. So remember, you have the gene, whether it's eye color, hair color, skin color, your height, the shape of your lips, the shape of your ears, the shape of your chin. But then you can have the different alleles. So remember what I compared the alleles to? It's like different flavors of ice cream. So remember, you can have the blue eyes, brown eyes, hazel eyes, gray eyes. You can have your blonde hair, red hair, brown hair, black hair. You can have your dark skin, light skin, and then everything in between all that. You can have the different heights, short, tall, super short, and everything in between that. So remember, and we know that the homologous chromosomes from what we talked about earlier are going to carry the alleles at the same location. Remember what that's called? Yes, the locus. Remember, it's the locus of the gene. So the maternal chromosome has a certain allele at the specific locus, and the paternal chromosome has a certain allele at the same exact locus. So we know that because what? We finished mapping the human genome in the year 2000, and then it was published in its entirety in the year 2003. We have been making modifications to it in the last 17 years in terms of the discovery of the diseases that basically the genes that cause the different and various diseases that you see. The second hypothesis that Mendel had was that for each character, an organism is going to inherit two alleles. And what does that mean? You're going to get one allele from your mom and one allele from your dad. And you can either have these identical alleles, which are homozygous, or you can have completely different alleles, which are heterozygous. So remember, these are terms that you're going to have to memorize and then explain in the final exam. Remember, the final exam is this Friday, okay? Remember that it is 100 questions, that it is completely multiple choice. It is online. You're going to require a lockdown browser, and you'll have it open from midnight on Friday morning until 11:59 Friday night. So you have all that time to take the exam. So basically, when he talked about the different homozygous and heterozygous traits, he came up with what's known as the law of independent assortment. And what he was able to determine was that alleles for different genes are sorted independently of one another. That means the genes are not linked. That's pretty extraordinary. So where do you think that would happen in an event or a process that we talked about earlier? Where would that occur? Think about that for a second. Yes, if you said meiosis, that is the process. What event then? What phase of meiosis? Metaphase 1 in meiosis 1. Remember that the chromosomes are going to align at the metaphasic plate, completely random and independent of any outside force. Nothing is controlling it. So that means if you have 23 chromosomes, that means you have uh, the possibility of 2 to the 23 possible arrangements. That means over 8 million, actually it's 8.2 million different combinations that you could have of just a single sex cell. That means a single egg or a single sperm. The chromosomes can line up in 8.2 million different ways. That's extraordinary. Why? You want variation. Why? You need variety in your species. Why? If we were all the same species, then we'd be susceptible to the same diseases, and obviously we would become extinct, because if we were going to have, for example, let's say that we did not have the ability to combat, talk about what's going on right now, COVID, right? What's going to happen with that? We would all be susceptible to it, and we would all die from COVID. All right. The third hypothesis that Mendel proposed is the fact that there are two alleles in the inherited pair that are going to differ from one another. And basically what's going to happen is you're going to have what's known as a dominant or a recessive trait. A dominant trait is what's going to be expressed. It's what you're going to be seeing. So every single trait about you obviously was dominant. That is what made you who you are. Again, we go through the basics, the most common: eye color, hair color, skin color, your height. Okay, those are things that you can see right away. And then obviously don't forget about the things that you don't see. Okay, are you susceptible to diseases? Can you combat certain diseases? What are the shape of your organs? What are the shape of everything that's internal, your different chemicals, molecules? All that is determined already at that time, so that was dominantly expressed. Recessive is what's not expressed, but it is carried within you. So you have the ability to pass that on to your offspring. So let's take a look at it. If we were looking at the possible pairing of homologous chromosomes, you could see all the different variations that could arise. So if you had homozygous dominant, you're going to have a yellow seed. If you had a heterozygous, and we know that yellow is dominant, that means you're going to still have the yellow seed. However, if you had homozygous recessive, that means you would have the green seeds. So again, it's about what is dominant and what is recessive. In the pea plants that Mendel was studying, the yellow seeds were dominant and the green seeds were recessive. Okay. The fourth hypothesis, again, this is all what Mendel determined. How? He had no computer. He had no calculator. Okay. No technology whatsoever. Okay. He was able to determine all of this basically by looking at his pea plants that he's growing in the garden, in the monastery in Austria. Okay, it's actually in Vienna, Austria, and it is open for tourists. Obviously not right now with everything that's going on in the world, but it is open for tourists, and you can actually walk around the garden and see the areas where Mendel actually stood and studied. So it's pretty extraordinary. For those of you that are interested in genetics, it is a fascinating trip. It's one I plan to take, hopefully, in my lifetime. So again, the law of segregation was Mendel's fourth hypothesis, and it stated that the sperm in the egg is only going to carry one allele for each of the inherited traits, because the allele pairs are going to separate, or in genetics terms, segregate from each other during the production of the gametes. How? During the process of, you guessed it, meiosis. Okay, so you can take a look at here, go back and review the lectures on meiosis, and you can see the maternal chromosome right here, paternal chromosome. Again, this is the chromatid after what? The S phase of the cell cycle. then you have the division, you have the sister chromatids, the homologous pairs. They will then separate, and then you have 4 haploid cells with different alleles on each of those different chromosomes. Remember, different alleles, but they're at the same location, locus. It's just going to have the different traits. So again, it's not going to be, there are no 2 sperm alike, and there are no 2 eggs alike. And remember, this is all before the process of fertilization even occurs. That means before the egg and the sperm meet to form the zygote. So again, extraordinary the randomness that will occur and the diversity that's created. That's why it's impossible for a couple to have 2 identical children with random and independent fertilization. Now, we're not talking about identical or monozygotic twins. That's a completely different event, which will be talked about later on. We are talking about random and independent fertilization. Okay, so keep that in mind. The experiment that Mendel conducted in the garden was a pretty straightforward experiment. It is pretty simple by our standards now, but you got to think that, I mean, imagine not having any of the technology that we have available right now, that he was able to come up with this. I mean, he was a genius. Remember, math was his forte. He could do anything with numbers. Okay, so what he did was he took the true breeding purple plant, which is described as homozygous dominant. So we got to use the correct terminology. Then you have the white flower, which is described as homozygous recessive. And he crossed those. When he crossed the homozygous dominant purple with the homozygous recessive white, in the first generation, he saw that the plants bloomed purple flowers. So his assumption was, okay, purple is darker than white. That means purple is always going to mask white because purple is a darker color. So he's thinking about things that he could see, the colors of houses, buildings, so on and so forth that he could see. So he was like, okay, he kept that. He had that theory for a long time. And he said, wait a minute, what happens if I cross to F1? So if I cross the 2 purple flowers from the F1 generations, he said, well, I'm going to get all purple flowers. He did that. And what was the result? Lo and behold, he saw 3 purple flowers and 1 white flower showed up. So he was like, aha. He had like an aha moment. And what was that? He said, wow, even though traits may not be expressed in a generation, they can be carried and expressed in later generations. Okay, so we have an example of that in my family. So 2 of my siblings, my brother and my sister, have hazel eyes, even though both my mom and my dad both have brown eyes. How did that happen? Well, it turns out my paternal grandfather had hazel eyes. My dad didn't get the brown eyes, but he gave it to 2 of his children. Pretty extraordinary. So it's exactly what we're seeing here. But as we're going to talk about later on, it's not as simple as 1 trait in 1 gene as it is in the pea plants. For eye color in humans, there is an estimate that it requires about 47 different genes that will determine the eye color of a single person. So it's not 1 gene like it is here. So what we're going to talk about that is it's known as multi-genetic or polygenetic inheritance. So we'll talk about that later on. So let's look at what we're seeing here. We have, using the Punnett square, so I know some of you are going to be familiar with the Punnett square from way back in high school. You have homozygous dominant, which is capital P, homozygous recessive, which is lowercase p, lowercase p. You cross those, and from his determination here, he was able to see that they were all heterozygous. Then he crossed the heterozygous, which is all of this right here, and he crossed the heterozygous, and he was able to come up with the fact that there was 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So the 3-to-1 ratio that we're seeing phenotypically, so we're going to talk about, remember, phenotype is what you can actually see. The genotype is the alleles. The phenotype is 3 purple flowering plants to 1 flowering plant. But the genotype is actually a 1 to 2-to-1 ratio. How? You have 1 homozygous dominant, 2 heterozygous, and then 1 homozygous recessive. Okay, so please review that. Practice Punnett squares. Again, you can just Google Punnett squares and actually go through in your textbook. And at the end of the chapter 15, there are problems, examples of performing Punnett square problems. All right, the dihybrid cross is a little more complicated. I don't want to get too much into it, but this is how he's able to determine that the law of independent assortment. And remember, the law of independent assortment stated that the alleles are going to segregate independently and random of anything else. So it's not going to be, again, one gene controlling another gene, and it's not required for one gene to control another gene in species. Okay, so you can just look at that in a dihybrid cross. And if you take a dihybrid cross, what you're going to be getting is the the heterozygous for the P shape and the P color. And you cross the heterozygous, and you always have a 9-to-3 to 3-to-1 ratio. So you can see that the heterozygous cross, with a heterozygous cross right here, you will have a 9-to-3 to 3-to-1 ratio for the F2 generation. That's all I want you to know about dihybrid crosses. I will not have any dihybrid cross problem on there. I probably won't even have any monohybrid crosses, but do practice them. I might give you one because you do have a minute to perform that on the exam, and it is pretty straightforward. All right, so the single gene traits that we have in humans, there are some, okay, AKA what we call Mendelian genes, are basically your cleft chin or non-cleft chin, your widow's peak or your straight hairline, your unattached or attached earlobes, and then your ability to be a tongue roller and a non-tongue roller. Those are all simple dominant recessive traits that are found in humans and us, and you could see those in people. And you've seen people probably be able to roll their tongue, and some people can't. That is all genetic, or the attached earlobe or the unattached earlobes. All right, disorders, a lot of the genetic diseases that occur, unfortunately, in humans are just single gene traits. So you're missing the proteins for those. You can have recessive disorders where you need to have both recessive alleles present in order to get the disease. The list of diseases you can see here: albinism, cystic fibrosis, phenylketonuria, PKU, sickle cell disease or sickle cell anemia, and Tay-Sachs disease. Again, you have to have the parents carry the recessive alleles, and both have to be expressed. If only one's expressed, you're not going to get the disease. You can be a carrier. If both are expressed, then you have the disease. So remember, physical expression in this case, unfortunately, means physical disorder. With dominant disorders, what you have is, if you have the dominant gene, you're going to have the disease. Dominant disorders include achondroplasia, which is dwarfism. We don't call it that anymore. It's not politically correct to call it that anymore. Huntington's disease or Huntington's chorea. Hypercholesterolemia, which is you're born with high levels of cholesterol. It doesn't matter how much how much food you eat or you don't eat, or how much fast food you eat or don't eat, or fatty food, so on and so forth. And polydactyly, multiple fingers. Obviously, if you look at your hands, fingers, you should all have 10 fingers and 10 toes. All right, various variations of inheritance. Again, this is after Mendel. So this is post-Mendelian genetics, okay? And we have multiple here. We have incomplete dominance, codominance, multiple allelism, the polygenic traits I talked about earlier, pleiotropy, gene linkage, and then environmental effects. So basically, with incomplete dominance, you have a heterozygote is the intermediate of the homozygotes. For example, in flowers such as carnations or snapdragons, you can cross a red flower with a white flower and you have a pink flower. It is not that you blended the colors. In life, in organisms, you cannot blend genes. Remember, the genes are individually expressed. However, there are certain species on the planet where you have an incomplete dominance, where you have an incomplete expression, and that's why you get the color pink for flowers. Codominance is where you have both of the homozygous traits expressed. A good example in humans is the AB blood type. So if you have type AB, that means you have the A and the B dominantly expressed. They're both dominant, so they're codominant. Co means together. So that's why you have the AB blood type. Multiple allelism, more than 2 alleles for a given gene. Again, the human blood type, the A, B, or O. For the A blood type, you can have 2 different alleles. You can have homozygous dominant, or you can have heterozygous. For the B blood type, you could have either homozygous dominant or heterozygous, and you'll still get the B blood type, or if you had A, you'd still have the A blood type. So again, multiple alleles. Polygenic traits. The polygenic traits, multiple genes are going to influence the phenotype. And again, what I talked about earlier, the eye color, the hair color, the skin color, height. And with the newer research, and we're talking about in the last 20 years, so this is after I studied and graduated medical school, again, remember the discussion, basically it's been discovered that there are several genes that account for the autism spectrum, the trait that causes a person to become autistic. So again, lots of study being done on that. We're not entirely sure about that yet. There's still a lot of research to be done on that, but that is something that's come out after the publication of the Human Genome Project. And there's a lot of work being done on that because we're seeing an exponential rise in autism in the United States of America, and we're really not sure what's attributing to that. There are a lot of theories. Some of them are out there, and then there are some that are plausible. Okay, so if you're interested in that, take a look at that. Pleiotropy is where one gene can influence multiple traits. Again, that's really, really cool. For example, sickle cell anemia. You can have sickle cell anemia, but sickle cell anemia, even though you have the disease, it's going to prevent you from getting a different disease. Does anybody know what that disease is? Possibly heard of it, maybe. Malaria. Yes, malaria. Actually, a sickle cell trait evolved as a mutation in mankind. Oh, I would say somewhere about 1 million to 2 million years ago. Because if the sickle cell trait did not arise, then we would have become extinct because we would have all been wiped out by the disease malaria. So basically, the origin of human beings in sub-Saharan Africa, there are a lot of the Anopheles mosquitoes. Anopheles mosquitoes carry the Plasmodium species. They are the species that are responsible for malaria. There's 4 different ones that if you're going to be taking micro, Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, and Plasmodium malariae. Those 4 different species of Plasmodium can actually be carried in the Anopheles mosquito. It bites you to take your blood, it stings you, and it's going to transfer while it's drawing and extracting your blood. The Plasmodium species is going to go into your bloodstream, and then it's going to go through its life cycle, and it's going to cause a malarial disease. If we did not have sickle cell anemia in mankind, we would have been wiped off the face of the earth about 1 million to 2 million years ago from estimation. All right, pretty extraordinary. So again, mutations give rise to diversity. So remember when I went over mutations, why are mutations so important? Yeah, we think about mutations as a bad thing, but in actuality, we wouldn't be here if it wasn't mutations, because remember, we came from a single organism, and that single organism was able to mutate and give rise to the billions of organisms that we find on our planet. Another important variation of inheritance is gene linkage, and basically that means that multiple genes are linked and travel together during the crossing over of meiosis. For example, a common one, again, I've known since I was a little kid, was seeing my friends with red hair and then freckles. So if you had red hair, you're automatically going to have freckles. That's because they are linked together. Those genes are found on the exact, those traits are found on the exact same genes. So that means if that gene for red hair is expressed, the gene for freckles is going to be expressed, and you're going to be seeing those. So again, really, really, really cool. Environmental effects that you can see, we know that temperature can affect the coat of different animals. So if you think about what a Himalayan rabbit or a Siamese cat looks like, depending on the temperature, or any of those species that can change their color to, again, to survive and to be almost camouflaged in their environment. That's due to the environmental changes due to the genes. So again, I went over to multiple allelism. You can look at that again. It's the example of the ABO blood type. And again, a gene can have more than 2 alleles. Pleiotropy, when genes influence the multiple unrelated traits, again, sickle cell trait and the malaria, and there's a parasite right there. Again, magnified, okay, 10,000 times, okay, the red blood cell right here, and you can actually see the Plasmodium species right there inside the red blood cell, destroying it, okay. The sex-linked traits, there are a lot of sex-linked disorders, and basically those are going to occur on what? Yep, the sex chromosomes, okay, whether it's going to be X or the Y chromosomes. A lot of times it's the X chromosome because the X chromosome is just another, it's the same size as the somatic chromosomes, so the autosomes, remember, so chromosomes 1 through 22. So there's a lot of information on the X chromosome. It can carry anywhere from 900 to 1400 genes, okay? And again, most of them are not involved in the determination of sex. What carries the sex determination is the Y chromosome. The Y chromosome has an area on it known, a gene or an allele, excuse me, known as the SRY gene. And the SRY gene has what's known as TDF. TDF is testes determining factor, and that's how you make a male in humans. So basically, the default sex for Homo sapiens sapiens, default sex is female. It's harder to make a male than it is to make a female in biology. So again, because it just normally wants to form the female species. It's just easier to do that. So then you have to change a law, introduce that gene to make the male in our species. For example, the difference between a man and a woman. All right. The Y chromosome, again, as little as 70, these are estimates, as little as 70 different genes compared to almost 1,400 genes on the X chromosome. It's a very, 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 very tiny, tiny, tiny gene, the Y chromosome. I'm sorry, the chromosome, the chromosome. It's really, really small, and it has only a certain limited number of genes. Again, testes determining factor, which is going to create testosterone and give you all the secondary sexual characteristics that make the man different from a woman and all that stuff. X inactivation is really an extraordinary process. I don't want to get into it too much right here, but I did want you to be aware of it. There are mammals that can undergo X inactivation, and again, it can occur. All right, and you can see it in different species such as the cats and the humans. All right, so I am going to stop here for now, and I will actually pick up in a little bit. So I will talk to you guys later on, and we'll wrap up this chapter.